Hey everyone, thanks for joining me today for episode 849 of People Behind the Science. I am excited to be resharing our interview with our guest, Dr. Joyce Ohm. Listeners, Joyce is exploring the epigenomics involved in development and in disease, particularly in cancer. How your cells package and store information in your genome to most efficiently use the genes they need is important during early development, but also there are problems with this process and the epigenome in diseases like cancer. And in our conversation, Joyce gives insight into her career, her work, and her life. So I hope you enjoy this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to the People Behind the Science podcast. Today, I am delighted to be speaking with our guest researcher, Dr. Joyce Ohm. So Joyce, welcome to our show today. How are you?
Thanks. I'm doing great. I appreciate it.
Well, I appreciate you taking time out of your busy day to tell us more about yourself and your experiences. But let me first start by telling our listeners a little bit about your current position and how you got there. So listeners, Joyce is an Associate Professor of Oncology in the Department of Cancer Genetics and Genomics at the Roswell Park Comprehensive Cancer Center. She was awarded her PhD in cancer biology from Vanderbilt University, and afterwards she conducted postdoctoral research in oncology at Johns Hopkins University School of Medicine. And prior to joining the faculty there at Roswell Park, Joyce served on the faculty at the University of North Dakota School of Medicine and Health Sciences. And today, Joyce, we are going to be talking both about your scientific side, but also about your life outside of science. So can you start by telling me and our listeners what you like to do with your time when you're not hard at work?
Well, mostly life outside of science these days has been riding my bike. So I love to do a lot of things, most of them outdoors. I like to hike and kayak, and I have two adorable furry little beasts that like to do those things with me. But I spent all of last week on my bike riding from New York City to Niagara Falls as part of the Empire State Ride. So 500 miles was a huge challenge because I'm not a cyclist in any way. So literally for the last 4 or 5 months, all I've done is work and cycle.
That is amazing. I can't imagine 500 miles. Were you going alone or were there others with you?
There were almost 200 of us. It's an official Roswell event that's in its 5th year now. And it's really, really kind of an amazing experience. Don't get me wrong. It was hard and we trained hard and it was hot and those hills hurt. But it's also just an extraordinary group of people. So scientists, people involved in the cancer world, lots of patients, survivors, family members out there all trying to raise money for cancer research. So it was a fantastic experience.
Well, we appreciate you making that arduous journey, and it sounds like a lot of fun as well.
It really was.
Well, Joyce, great to hear a little bit about life outside of science and how you stay active, how you spend your time. But I'd love to talk about your research as well. And we hinted at when we talked about your work there at Roswell Park Comprehensive Cancer Center that you do work on cancer. But how do you describe the specific research you do to people outside the field?
Well, it's a struggle, and I know a lot of the scientists listening probably feel the same way. I think for years after I got my PhD, my family still didn't understand that I couldn't write them prescriptions and I didn't treat patients, and that's not what I did. So you try over the years to figure out how you're going to explain to people that you're in the lab behind the scenes. The way I do it for new students coming in is I tell them that you're going to do something that no one's ever done before. And in fact, in the research lab, we don't ever do anything that anyone has ever done before. So you're kind of trying to find your way on a journey without a map. And you pull in the information from all kinds of different sources in the hopes that we're all going to get to a place at the end where actually we're making a difference for patients. At least in the cancer field, that's always our end goal, is to try and keep things very relevant for the patients that we're hoping to treat.
Absolutely. And maybe more specifically, you work on epigenomics. So can you talk a little bit about this?
Absolutely. So epigenetics was a new field as I was finishing my graduate work. I mean, it had been around for a while, but it was really kind of hitting the mainstream and becoming really, really popular. So I always describe epigenetics as the way your cells pack away their genome. So if your genome is everything you own in the world, then your epigenome is how you pack for that journey. And every cell in your body has to decide what tools it's going to need at the ready and what things it maybe doesn't need so much. And so it has to decide which of those 25,000 genes are going to be accessible and which ones aren't. So it can pack them away in ways where it can use them most efficiently. It's a key component of development. It's absolutely essential as different cells decide what they're going to be in life. And the epigenome is one of the things that gets just royally screwed up in cancer. I always tell people, if anybody's got a 2-year-old out there and you let them pack for a vacation, that's very much how cancer cells package up their epigenome. You may not know what you're going to end up with in the end.
I love that analogy. Mostly snacks and stuffed animals, right?
Probably.
Well, wonderful to hear more about your research. And I look forward to chatting about some of these specific projects that you're working on as we go through our interview today. But let's pause for a moment to talk about what motivates you. I think this field of cancer research is so exciting to be a part of. But do you have a specific quote or sayings or forces that motivate you to do the work you do?
I have a hard time taking most of this too seriously. I think my favorite has always been the Einstein quote, "Only two things are infinite, the universe and human stupidity." And I'm not so sure about the former. It's very true. And in today's world, we kind of have to laugh a little bit about that.
Absolutely. And I think it's particularly applicable to science because we're not immune to these sort of stupid human mistakes, right, as scientists?
No, we make them every day. And in fact, it's a huge part of what we do is making mistakes and going back and doing things over and laughing at yourself a little bit. I always am surprised when the students come in and they'll be really upset because they did something dumb in the lab. And the best thing to do is say, hey, been there, done it. We've all done these things. Mm-hmm. So go do it again.
Yep, exactly. Get back on that horse.
Yep.
Well, I love that quote. And I think it's so important to talk about what motivates scientists, and not only the forces and the quotes and the sayings and the mantras that keep scientists going, but also the people who've been hugely instrumental on your journey. So Joyce, when you think back over your own experiences, are there particular people who rise to the top as mentors or role models or just people you looked up to?
The top of the heap will always have to be my graduate mentor. So I did my PhD with a really, really talented physician scientist by the name of David Carbone. He was at Vanderbilt then. He's now at Ohio State University. And when I started graduate school, I knew I wanted to do cancer research. I knew that was the direction I was headed in, but that was still a really vague thing. And I didn't really know exactly specifically what I wanted to work on. And he really helped me dive into that in terms of where did I want to go? Where were my real interests? He gave me almost unlimited freedom to ask questions and to think about the problems in front of me and really work through those. And it was amazing. He's a crazy talented physician. He's a great dad. He's a good man. And he has been someone who's been supportive of my career ever since. And I will always be grateful for that. The other thing he really did for me was because he's a physician scientist, so he was seeing patients, but also in the lab, he really helped hone my training so that everything we did was translational and kept us out of the weeds a little bit. And anytime we got a little bit too much into the science details, he'd remind us that we were working for the patients there. And that's something that has stuck with me through my career. It's something I try and instill in the people in my lab now, is to kind of bounce back and forth between the minutia and the details of the experiment in front of you and that big picture. And I will always be grateful to him for that.
Absolutely. Well, it sounds like you had a phenomenal mentor in graduate school. And you mentioned that already at that point, you knew you wanted to do cancer research. So Joyce, let's rewind even farther back and start talking about how you got interested in science and maybe cancer specifically in the first place.
Well, that was very much life. So I lost my mom at a very young age to a rare form of cancer called clear cell sarcoma. Therapy probably would have been cheaper and easier. Instead, I decided to get a PhD, and that was what I was going to do. And I was going to kind of spend my time seeing if I could make a difference in that area. And it's been really great because one of the things you learn when you do this is that people will come to you. So now I can help. Now I have answers. Now I can at least point people in the right direction. Now I know how to dig in and do the research and give people the answers that I didn't have at that point. So that's been a really powerful motivator for me. And it's one that's continued because of wonderful interactions with friends and family members and friends of friends who are going through this really, really difficult struggle and really just want information. So it's a really wonderful, positive feeling to at least be able to give a little back in that area.
Absolutely. Well, Joyce, I'm so sorry to hear about the loss of your mother. And it seems like so many of the people I know, and myself included, have been touched in some way by cancer with the loss of a loved one or a family member or someone who is currently fighting the fight. So we really appreciate the work you do. And I'm curious, what turned your eye towards research as opposed to medicine?
It's a little bit of a funny story because I really was going to go to medical school. I went to a smaller state school in Michigan for my undergrad. And my only exposure to research in those years was a summer project that I did looking at the mating habits of tree swallows.
That wasn't where I thought this was going, but continue.
No, I think that was really kind of the one experience that I had open to me. And I did it. And I spent hours sitting in a field, waiting for birds to fly into bird boxes. And then we would do bacterial swabs. And it was fascinating and interesting and completely not what I wanted to do. So I was headed to medical school and thought that that was what I wanted to do. And graduate school was going to be my backup. That was going to be the thing that I was going to do when I didn't get into medical school or thought I might not get in. And it was very similar timing. I had medical school secondary apps in front of me, and I had all of my graduate school applications in front of me. I'm sure many have been through this. The med school secondary apps are a lot of questions about why you want to be a doctor and what you're going to do there. And your grad school applications are similar. Why do you want to do this? And I struggled through those med school applications. They sat for weeks because I couldn't really articulate why I wanted to be a physician. And so again, in my backup plan, I pulled out my grad school applications and I started to write about research and what I was interested in and why I wanted to do that. And they flowed beautifully. And it took me a couple hours and some polishing and editing, and they were all done. And I went back to those med school apps and I thought about what I'd seen and what a physician does every day. And I didn't want to do it. And it became very clear through that strange exercise that we all have to do and no one wants to do that I was probably supposed to go to graduate school. So I actually pulled my med school apps and I went and became a graduate student.
Well, that is awesome. Joyce, I'm glad you took that extra moment to stop and reflect, because I feel like that period where you're applying for these things is kind of just like a frantic push to get everything done. So for our listeners out there who are going through this struggle themselves of trying to decide between the grad school and the medical school opportunities, I think that's a great place to stop and think about, why am I really doing this? And maybe which one do I have a bigger passion for? So that is awesome. So then can you tell us about your graduate school experience once you made that decision to follow that path?
Sure. So I was accepted to a variety of schools. I actually chose Vanderbilt for many reasons. One of them being at the time, it was one of the first integrated programs. So the IGP integrated graduate program there had a common first year for all departments. And of course, now that's very common. In fact, it's the norm, I would say, where you're actually going into kind of an umbrella program and you're going to do rotations and get a feel for where your interests are and what you really wanted to do. But at the time, it was still pretty new. And it was the main reason I chose it is I wasn't exactly sure what area, other than I was pretty sure I was going to go into cancer research. So I took advantage of that. It was an interesting experience. I struggled a little bit through my first year classes and especially through my rotations. I was one of those students who did my 3 rotations and had not found a research home. Mm-hmm. None of the labs were quite a right fit. I wasn't sure what I was going to do. I found myself questioning again, is this the right thing for me? And one of my friends at the time had just finished a rotation with David, who ended up being my mentor, and said, this is the perfect lab for you. So I went to him. I said, listen, I want to do a 4th rotation. Will you take me? And he said, yes. So it became apparent very, very quickly that actually it was a perfect fit for me. So I'm glad I didn't settle into one of the first 3 choices that I thought were all going to be great, because it ended up leading me to where I was probably supposed to be.
Definitely. Well, I'm glad you shared the story. I actually ended up doing 5 rotations myself.
It's not just us. There's lots of us who have a hard time finding the right fit.
Exactly. For me, actually, I kind of knew the lab I wanted, but they didn't necessarily know about funding until after I had completed that 5th rotation. I was like, oh, good, this worked out. So I was on my way to a 6th before that decision came into play. So well, Joyce, it's great to hear about how you found this perfect fit in grad school. And I'm glad that you did, because I think finding the right mentor can make all the difference in your graduate school experience. So then can you talk about what happened next?
Sure. So the lab I ended up in was doing something, again, I didn't actually think that I was going to do. It was very much immunology focused. And so at the time, David had a really novel observation that tumors secrete various factors, including vascular endothelial growth factor, or VEGF, and that these factors can actually induce immune suppression and help tumors evade immune surveillance. So they had recently found that VEGF could inhibit dendritic cell differentiation and form these immature dendritic cells that now play a very, very key role in modulating tumor immunology. And they had also noticed that the T cells were depleted as well. So literally that was my graduate project. I was in his office. He said, Joyce, go figure out what happened to the T cells.
Oh, cool.
So I spent 5.5 years figuring out what happens to the T cells and how tumors actually can help inhibit T cell development as well and modulate this overall immune response. And at the time, it was fascinating. We worked through a lot of problems. Nothing worked. It was very much a trial and error time in my career where we were just eliminating possibilities. And I remember early on, I had drawn out in chalk on a chalkboard, 3 chalkboards, the whole pathway of T cell development and all the checkpoints and places where things could go right or wrong, all the decision points. And I worked my way back through those over the course of the last few years. And it was really one of the last ones that we looked at that ended up being kind of the point where these factors could inhibit it. It's really cool, though, because at the time we knew immunotherapy should work. We knew that it could work, but we couldn't ever make it work. So it's funny to me now, 15 years later, all of that work is actually some of the work that's getting cited more than anything else because now immunotherapy is such a hot topic in the cancer world. So they're going back and trying to look for ways to improve it. It's been a nice reaffirmation and lesson for me in remembering that we don't actually always know the impact of what we're going to do when we're doing it. And sometimes you have to wait a really long time, but it's kind of a blessing to watch those things develop over time and think that maybe you had a little tiny part in it.
Absolutely. Well, it sounds like your graduate work is coming back to have a big impact. Can you talk next about how you made that decision about where to go for your postdoc?
I can. I'm starting to feel like these stories are going to make me look bad over time because they're all a little bit random. I tend to be a little bit impulsive, and this one isn't not going to help that image. So I was at an AACR meeting, must have been like my 4th year or kind of into my 5th year of graduate school. And lots of scientists have been to these big meetings. You know that there's rooms and rooms and rooms full of talks. And I was waiting for a talk I wanted to see a couple hours later and kind of killing time, wandered into a symposium on epigenetics. Didn't really know what it was at the time, other than kind of a basic understanding. And Steve Balin, who ultimately ended up being my postdoctoral mentor, was talking, and several other really prominent people at kind of the beginning of this really big boon in the field of cancer epigenetics. So I literally sat down to kill a little time, and I ended up spending the whole day missing the talk that I was going to go to.
Oh, wow.
And spent most of the next day searching out epigenetics talks around AACR and seeing what else I could find. And Steve came a couple months later and gave a seminar at Vanderbilt. And I aggressively pursued a postdoc with him because it was really very much what I wanted to do. One of the things that we'd found kind of in the immunology work was that tumors could block that differentiation by working on stem cells. And I just had a feeling and knew from what I was starting to hear that that repackaging of the epigenome into a stem-like state probably played a big role in tumorigenesis. And Steve was fantastic, and he let me run with that. And so I very much had that experience as a postdoc also, where I could kind of ask an interesting question and he let me go. And that was it. It was a little bit random. And then I just kind of fell in love.
I love it. Well, Joyce, this story is phenomenal. And I particularly like hearing these stories of how elements of serendipity kind of help guide scientists to where they ultimately end up. And I think this is a fabulous example of that. So can you talk a little bit about your current research now, and what is a project that you have going on that you are just so excited to be working on?
Sure. So again, a little bit of serendipity, but my work has gone almost completely full circle back to soft tissue sarcomas, which is the kind of cancer that my mom had, but it wasn't an intentional move. While I was at Hopkins, the whole induced pluripotent stem cells story came out. So this idea that you can reprogram a terminally differentiated cell back to an embryonic-like state. And we already knew at that point from a lot of Bob Weinberg's work and the work of others that many of the most aggressive human cancers have an embryonic stem cell-like signature attached to them. So while lots and lots of scientists were kind of jumping on the iPSC bandwagon to see if they could use these cells for regenerative medicine, we were interested in them for another reason. And we were interested because we thought this might actually be a perfect model system that we could use to study induced dedifferentiation and how cells might regain an embryonic-like state. And those early IPS stories were interesting because while they could get cells back to an embryonic-like state, what they talked a lot less about was that that was a really, really rare event. So we teamed up with Dr. Lenzao Cheng, Dr. Prashant Mali, who's now at University of California, San Diego, and they were working with iPSCs And what we said is, hey, you guys take the ones that go all the way back that you're going to use for regenerative medicine. Can we look at the ones that don't completely reprogram? Because our hypothesis was that those cells would actually be very cancer-like. And it turned out to be true. The iPS clones that don't make it all the way back or make it even almost all the way back have really remarkable features of malignancy. High mitotic indices. They have areas of focal necrosis when you put them into a mouse. And they're really, really very confused cells with a messed up epigenome. And that's exactly what we kind of thought might happen. But one of the things that popped out of that was one of the tumors that we grew from these failed iPSC cells was a small, round blue cell tumor of undifferentiated origin. And David Berman was the pathologist on the project. When I took it to him, he said, hey, Joyce, this looks like a small cell lung cancer or a small cell pancreatic cancer. And then I went to Jim Herman, who was the med onc, and I said, Jim, do we ever see these in the clinic? And he said, yeah, these undifferentiated small round blue cell tumors tend to be sarcomas or are treated like sarcomas. And I didn't work on sarcomas at all. All of my training up until that point had been in lung cancer, but I realized I'd better learn something about these small round blue cell tumors. And shockingly enough, what pops out is this whole class of rare soft tissue sarcomas with this small round blue cell phenotype. They predominantly affect children and young adults. And one of them actually is the subtype of cancer that my mom had had. And they're all translocation driven. So these are really, really unusual tumors when you compare them to kind of the more common cancers that we talk about. They generally have one predominant mutation, this translocation. They have few other mutations. In fact, their mutational burdens are very, very low. And often the fusions involve proteins that regulate the epigenome and control epigenetic remodeling and stemness. So I found myself in this very serendipitous area where everything I had worked on to date really started to kind of fall into place. So at that point, we really changed. The entire focus of the lab now is on understanding how these fusion proteins and their reciprocal fusions, which we think function as long non-coding RNAs, reprogram the epigenome in these cancers. And can we exploit that? Can we develop new therapies for these? Because these are cancers that we have very, very few therapeutic options for. If you can get them out surgically, the patients do great. If not, they tend not to respond to chemotherapy or radiation. So we really, really do need targeted therapies to do that. So that's very much what we're working on now. It's been a fun journey.
Gotcha. So do you have any ideas or thoughts on what some of the potential targets might be for this kind of cancer, or is it not quite at that stage yet?
We've started a couple pieces of this. And I think what we're looking at is combinations of epigenetic remodeling drugs. So hypomethylating agents, HDAC inhibitors, the EZH2 inhibitors, all may actually be interesting. But I have a crazy talented grad student right now who is running with a really fascinating project showing that these soft tissue sarcomas actually have very messed up systems in terms of both replication and transcriptional stress. So he's starting to look at things like PARP inhibitors in them, and those are great drugs for us because they're approved for other cancers, but haven't really been tested in this population. And yet these cells are exquisitely sensitive to some of those. And so for us, that's going to be a lot of fun. The other piece is we've had to learn how to be RNA biologists because the tumors actually have lots and lots and lots of non-coding RNA fusions in them that we really don't understand what they do yet, but they're there. And we're hoping that we can exploit that a little bit as potential therapies down the road.
Absolutely. Well, Joyce, this is fascinating work. And I know me and our listeners wish you the best on these projects. And I know as a scientist, there are these exciting moments of success or just excitement going in to do the work every day with that big goal in mind. But I know the life of the scientist is also full of challenges and struggles and down days as well. And we want to make sure that our listeners are aware of that and kind of have a sense of the challenges that scientists face. So do you have a story of your own of a challenge or a failure that you've been through that you can kind of walk us through and tell us how you made it through?
Yeah, the worst of them was about a 6 to 9 month failure that I struggled with towards the end of graduate school. And I was ABD, so all but dissertation at that point. My work in the lab was done, and David had asked me to work on a patient sample that had come in. And this was a really, really unusual patient with a stage 4 bronchiovelar carcinoma who was one of the first patients to be treated on the EGFR RTKI. And the patient was a super responder, had done amazingly before relapsing and eventually losing their battle with that cancer. And so we were trying to figure out what had happened in the tumor to make them no longer responsive to that therapy. I had a post-autopsy sample in my hands, and we were looking for mutations in the tumor. And it started with a success. We found a mutation and thought that we were on the right track. And then we pulled the original patient samples, and it turned out the mutation was already in there. And anytime that happens, the first thought as a scientist is, what did I do wrong?
Right.
Did I mix up the samples? Did I contaminate the samples? So I did it over and over and over and over again. And then I went to Dave and I said, listen, I think this is really in here. And he said, nope, you screwed up.
Oh,
Go
no.
do it again and again and again. And we spent about 6 to 9 months trying to make sure that the experiments were done correctly and that we hadn't made a mistake, because it was important. And we had to understand this mutation, whether it was there or not. We eventually took the extreme measures of like a clean room environment. We had new samples shipped to a lab on another floor that had never seen the tumors, different pipettes, different reagents, different everything, and eventually found that the mutation was in the original tumor. And it was a failure in every way except the real way, because it turned out that that was actually the best possible scenario, because we could rethink our hypothesis. And the mutation wasn't actually driving the relapse. The mutation, it turns out, was driving original response to therapy. So now, again, 15 years later, non-small cell lung cancer patients get screened for EGFR mutations now. Then they go on these special targeted therapies. But at the time, I wanted to quit and you wanted to stop. And I was sure that we had just screwed up somehow. And those failures are hard when the gel won't run and the PCR won't work and your sequencing comes back and it's a mess and you can't even read it. And it's day after day after day of kind of beating yourself up. And the strange part is that's all perfectly normal for our job.
Right.
So that's a hard one to explain. And so I have kind of a can talk that I always give the grad students. They get one when they start in the lab. They get one the first time they show up in my office in tears. And that always happens. And it's usually a version of that story because they're just so frustrated. And it's really hard to fail at something and make mistakes over and over and over again. But you pull out of it eventually.
Yeah. And I think in this case, particularly because you were so sure you were wrong, you just kept searching for evidence that there was a mistake and there hadn't been.
Correct. That's the easiest explanation, right? We all kind of suffer from that confirmation bias. And when you think your hypothesis is right and your data is conflicting with that, it's easier to assume you did something wrong than to accept that maybe the hypothesis was wrong.
Absolutely. So when you were nearing month 9, perhaps, what did end up keeping you going, Joyce, and keep you from quitting?
Probably just sheer stubbornness. I think it's a very common trait among scientists also. There's just that feeling, I will get this right and I'm going to do this. And you've got your people around you that kind of keep you going. And I definitely relied on those. I had good friends that said, get back in there, because there's days when you don't want to.
Absolutely. Well, thank you so much for sharing this story with all of our listeners out there today. But Joyce, we don't just want to talk about the bad days. I'd love to talk about some of your successes as well. So do you have a story of success, whether it was a huge win or even just a small victory that was particularly meaningful, that you want to share with us today?
My favorite one is actually super recent, and it's a failure, but it's a huge victory for us. And it has to do with the sarcomas that we're working on right now and these really strange non-coding RNA fusions that we found in them. Because it took us months to even convince ourselves that they were even real and weren't some sort of strange sequencing artifact. And we sent the paper out, and then we wrote a couple of R03s, so little grants, to just kind of test the waters in this area. And I called everyone I knew who worked on non-coding RNAs or RNA biology, and I was like, have you ever seen these? What do you think they are? And virtually every one of them was like, well, it's probably an artifact, but it's cool. So we sent these R03s in. Neither of them got funded, but they both got discussed and they both got scored pretty well. And nobody told me I was stupid.
That's a victory right there. Yeah.
I know it sounds silly, but it was a huge victory, because when you're venturing out of your field like that and you don't know what you're going to get, just sometimes someone not saying, hey, you're an idiot, is a huge win.
Yeah, I think putting those ideas out there and having people kind of seriously consider it and give you that feedback is really validating.
Yeah, absolutely. So I'm totally celebrating that right now. Of course, we have a lot of work to do and we'll get those grants back in, but that's our most recent success and it's actually felt awesome.
Well, congratulations, Joyce. That is to be celebrated for sure. So does your lab have a particularly favorite way that they celebrate successes?
We always end up out somewhere. Western New York is beautiful. There's lots of places to go and explore. There's usually cocktails involved. I'd be lying if I said that there wasn't. But we haven't found our coolest spot yet. Niagara Falls is close. We have done that a handful of times. And at least when new people come in, that's probably the most fun place to take them.
That is awesome. Do you do the boat rides and stuff there?
We do. That's one of the things I did with kind of my first bunch here is did Maid of the Mist with them. And they had a blast. So especially people kind of coming from all over the world that have never been. And so it's nice that you have something that cool in your backyard. They want to do the speedboats where you go down under the rapids. And I haven't done that with them yet.
It sounds a little maybe too intense for me. So
They say the boat fills up. And I think that's the main reason I haven't done it. The boat kind of half fills up with water, up to your waist when you dip under the rapids, and I'm working my way up to it.
Right. Building the courage.
Yeah.
I love it. Well, congratulations again on this success. And we talked about life outside the lab a bit in terms of what you like to do together outside the lab. And one of the things that I also love chatting with guests about is what they are reading, whether it's related to science or not. So Joyce, do you have a book that you can recommend to me and our listeners that you've really enjoyed?
So here's my problem. My favorite book is actually out of print. And I had to scrounge for a used beat up paperback copy years ago because I had read it a long time ago. It's a book called Amamori. I don't know who the author is. It has nothing to do with science. It's multi-generational historical fiction with some love story popped into it. It's completely escapist, but rich in historical details. And it kind of goes through Japan and the silk trade in World War II and how silk was used in the parachutes in both the German army and the Japanese army. And it's just a complete escape from what I do daily. I have a lot of friends who read science books at night. I am not one of them. I read a ton and it's always fiction. Sometimes the more mindless, the better, because I just want to check out for a little while.
I completely understand. I read a wide mix. Sometimes science books, sometimes something completely fiction or fantasy. So I completely understand. And we will add Amamori to our website for our listeners to find there. They will have to do some serious digging, it sounds like, if they want to get their own copy of it.
They will. I don't even know where you'd find one now.
But listeners, it sounds like it's worth it. So, well, Joyce, we've talked about some different aspects of your life and of your science as well. And one of the things we haven't really touched on are some of the opportunities you've had for travel. You touched on some of the conferences you've been to and the great experiences you've had there, particularly this introduction to epigenetics through a conference. But do you have a particular place that your science has taken you that's just been the most memorable?
It is a huge perk of our job. If there's listeners out there who are seriously thinking about a research career, know that you can see the world and you can check different places out. Just this past fall, I was in Italy. I was in China. That was amazing to give a talk, an invited lecture there. I spent some time in Hong Kong. I've been to Hawaii. I've been to Portugal. But probably my favorite was India. So I gave a talk in India at the Indian Science Congress a couple of years ago with a couple of my colleagues from the University of North Dakota. And it was absolutely extraordinary. We flew into Delhi. We made our way down to Bangalore. The conference was actually in Mysuru. And then back up, we took an extra couple days. Of course, you should always do that when you can. And went to Agra and saw the Taj Mahal. And we did spice markets in Delhi. And it was extraordinary. The people, the food, the smells was all just a really, really powerful experience there. And I'm headed back to Italy this fall. I have to pick a couple of cities to bookend that trip because the conference I'm speaking at is on one of the islands in Italy. And so I have to pick a couple European cities to bookend that trip. So if people have suggestions, you should send them to me.
Yeah. Do you have any front runners at this point?
The front runners are probably Madrid and Berlin. Madrid, because many friends have told me it's absolutely a must-see. And Berlin, because it's one of the German cities that I can fly to the conference from. And the conference overlaps with Oktoberfest. So maybe a German stop makes sense.
Good timing. Yes, of course. I love it. Well, it sounds like you've had some phenomenal travel experiences. And I think you're absolutely right. For a lot of people out there, they may not realize that this is part of the career as a scientist, sharing your work with people from around the world, traveling to these conferences to talk to people. So I think it is one of the perhaps unknown perks of life as a scientist. And in addition to the great places you're able to go, you're able to work with some phenomenal people as well. And I think this maybe goes contrary to a lot of the stereotypes out there about scientists, but most of the scientists I know are just fun, creative, amazing people, and funny things happen when we get together. So, Joyce, do you have stories of some of the quirky traditions or funny memories that you've experienced with colleagues that go contrary to some of the stereotypes about scientists?
It kind of cracks me up, but if I had $1 for every time I met someone who said, you don't look like a scientist or you don't seem like a scientist, I could probably retire. It happened probably 15 times just this past week on this bike ride.
Oh, wow.
Because there's a stereotype and I don't fit it. And very few of the people I know fit it. So I'm still not sure exactly where it comes from.
Mm-hmm.
We used to have a blast. We still do. Some of the quirky lab traditions you're probably not supposed to do anymore. So everything from scaring your benchmate with dry ice bombs is really fun. And we used to make pipette dart guns out of the plastic pipettes and then use the tips to shoot them. And we'd set up a big target at the end of the lab and play a little bit there.
Mm-hmm.
I was at a conference once and there was a woman there with the light up hula hoop who spent like most of the evenings doing glow in the dark hula hooping for people. And it was hilarious.
Did you try it?
I did not.
No, I was going to say, I'm not sure I could make it.
So it just, you're right. Silly, fun, creative people, a lot of outdoorsy people. That's kind of where we tend to go. A lot of hiking, kayaking, biking, all those things are things that I do with my science friends. And we all kind of seem to gravitate to that kind of a world.
Absolutely. And it sounds like this recent Empire State bike ride that you did was maybe a tradition for the Roswell Park community there. So do you guys have other traditions currently that you engage in?
So Roswell Park has kind of 3 main fundraising events. They do the Ride for Roswell, which is one of the largest single day cycling events in the country. And it has about 8,000 riders. And you can ride from between 3 to 100 miles on that day. They have the 11-day power play. This is Western New York. We are all about hockey here. So the 11-day power play is about 2,500 hockey players this year playing round the clock for 11 days.
Oh, wow.
So they take shifts on the ice. And then the Empire State Ride, which of course I've fallen madly in love with and will probably have to do every year from now on to bike across the state. So those are kind of our big ones here at Roswell. I'm sure other places have kind of similar traditions and similar ways to bring people together.
Gotcha. Now, do you anticipate you'll participate in the hockey event?
So I really am a terrible ice skater, so probably not. I grew up in Michigan, but I never learned. So me on ice skates is bad enough. Me on ice skates with a hockey stick is probably not anywhere in my immediate.
It's a danger to yourself and others, right?
Absolutely. I will happily cheer on the more graceful, talented colleagues and friends that I have.
I love it. Well, Joyce, it sounds like you have wonderful traditions there at your current institution, that you've had some great experiences in the lab over the years. And you're absolutely right that most of the people that you meet in science don't really meet this stereotype. I think, like we mentioned, you're creative and you're working on really innovative things, and you sort of have to be creative and not take yourself too seriously to be successful in science and to answer some of the big questions that you're answering. And I'm curious, if we took away all the restrictions that you normally face as you answer questions in science, things like funding or staff or technology or feasibility limitations, Joyce, what is the one question that you would want to answer most?
Honestly, it's the one we're working on right now. Maybe that's cheating a little bit, but I'm actually doing exactly what I want to do right now in terms of research. And I really, really want to understand where these translocations come from and how they kind of systematically and methodically change these cells and transform them. So I guess it's my job to make sure that funding and staff and technology are not the roadblock.
Absolutely. So if we did give you everything you could dream of today, how would you enhance your current program?
You know what I would love to do? And I've always said this, if I ever won the lottery, I would form my own institute and I would bring in people that you've worked with. And I could probably name 15 or 20 of them over the last couple decades that I've been in this business. And I won't name them because then I'll leave someone out and I'll get in trouble. But there are some crazy, brilliant, outside the box thinkers, just like you said, creative types that I would love to put into like a think tank. And I think if we did it, we could solve these problems.
Very cool. We'll get those lottery numbers in and we'll keep our fingers crossed for you.
Yeah. I need a lot of money to do that though.
Absolutely. Well, it sounds like a wonderful endeavor and maybe a quick way to get to a solution to some of these problems that it's just, they're so massive and there's so much work required to get the answers that you need that it's going to be a little bit of a wait for us all. But I know you've shared so many great insights with me and our listeners throughout our conversation today, and so many stories from your own experiences. And we'd love to ask you for one more thing here at the end of our program, Joyce, and that is some advice. So if you think back over your life and career, is there one piece of advice that really rises to the top as something that helped you on your own journey that you can tell us today?
When I was trying to decide between graduate schools, I was on the phone with one of the faculty members at one of the places I didn't choose, just for the record. And I asked him, I said, how do you make a decision like this? Because you don't really know what you're doing or where you're going, and you're kind of trying to guess for the future. And what he said to me was that you flip a coin. And I kind of balked a little. I'm like, really? That's the most ridiculous advice I've ever been given. Mm-hmm. And then he said, no, wait, you flip a coin. You don't do what the coin tells you to do. When you see that heads or tails, you listen to whatever your gut's saying. And it's true. You flip a coin, and there's that moment when you first see it where it's either a moment of confirmation or like, ooh, maybe I should do best 2 out of 3. And he said, that's the voice you listen to. And I've used that over my career. I've shared that over my career. And I really think it works, because I think as long as you're trusting your gut, our brains are pretty amazing and they can process things maybe if we haven't really thought them through. But that hasn't steered me wrong yet. And then I guess the piece of that then is then you embrace your choice. Because with the right kind of attitude, I think I could be happy at a lot of different places and a lot of different choices. And I think you go all in from there. But yeah, flip a coin and trust whatever your gut tells you about the results.
I like it. That's great advice. I'm going to have to start keeping a coin in my office. Make those difficult decisions for me. Well, Joyce, wonderful advice for our listeners. And I think that particular piece of it, to kind of be decisive, to embrace that decision and go with it, is really important, because you do sort of get this decision paralysis sometimes where you hem and haw for so long that you really just need to pick something and go with it and trust that something good's going to happen.
Yep, absolutely.
Well, wonderful advice for our listeners. Is there any other last piece of advice you want to share with us, or any last note of inspiration you'd like to end our conversation on today?
Just go with it. Dive in. If this is something that you're thinking about or something you feel like you could be passionate about, give it a shot. The worst thing that's going to happen is you'll find out it won't be for you. But I think for many of us, a research career has been very much a labor of love. And I've been very fortunate in my career. So dive in and see what's out there, because you never know what you might be able to contribute.
Wonderful message to share with our listeners. Joyce, if they want to learn more about you and the wonderful work that you're doing, where should they go, or how should they get in touch?
You can always email me at joyce.ohm@roswellpark.org. I'm on Twitter, although I'm not a super good Twitter member, at Epigenomics, with Ohm being spelled O-H-M like my last name.
I love the pun.
And I'm easy to find.
Fantastic. Well, listeners, follow Joyce on Twitter. Get in touch if you have questions. And Joyce, thank you so much for sharing a piece of your story with all of us today.
Thank you. It was my pleasure.
Well, it was a delight to chat with you, and listeners, great to have you here as well. We'll see you next time on another episode of People Behind the Science.